All right. Today we're welcoming Jacob Becraft, who is a synthetic biologist and biotech entrepreneur, as well as the co-founder and CEO of Strand Therapeutics. Strand is situated at the intersection of synthetic biology, immunotherapy, and mRNA therapeutics. Jake started his career developing the Safer platform at MIT, the world's first synthetic biology programming language for mRNA. He is an inventor on a multitude of patents in the biotech and gene therapy space and has won numerous awards for his scientific and entrepreneurial achievements, including a Goldwater Scholarship, a Vertibi Fellowship, and an Amgen Fellowship. He is an advocate among the life science entrepreneurial ecosystem for supporting founder-driven biotech startups and works to help increase young technical founders in biotech entrepreneurship. Jake also serves on the executive board of Public Health United, a nonprofit focusing on educating scientists across disciplines to foster their communication skills in order to become their own scientific ambassador. He received his PhD in biological engineering and synthetic biology from MIT and his BS in chemical and biomolecular engineering from the University of Illinois at Urbana-Champaign, graduating magna cum laude with distinction. In his free time, he also is an avid backcountry snowboarder and trains Brazilian Jiu-Jitsu. So thank you, Jake, for joining us today. We're thrilled to have you. And also welcome to all of the Nucleate Insights cohort for our second session. We're so excited to kick this off. And thank you, Bowen, for letting us kind of use your PowerPoint and helping us with throughout the presentation today. Jake, do you mind kicking things off today with a brief background on how this research got started that ultimately led to the formation of Strand?
Sure. Yeah. Thanks, Rebecca. I was just thinking that my biography is way too long as we were doing that. I think we should really simplify that down. Yeah, great to be here, you guys. Thanks for having me. So I am a scientist, and I run this company called Strand, some RNA company that Rebecca just gave a lot of information about. So background on the tech was going back around 10 years when I was starting, when I came out to MIT to do my PhD. Actually, I guess my motivations probably go back a few years before that when I was at University of Illinois and I was working on DNA delivery particles. And one of the big problems with DNA delivery particles and DNA, synthetic DNA as a therapeutic modality is getting into the nucleus. The nucleus is like this, very well guarded and frustrating thing to penetrate. And in order to get DNA to be active and to transcribe and translate the genes you encode, you have to get into the nucleus because that's where all the transcription machinery is, of course. And so I decided that I was going to go into synthetic biology actually because I wanted to ignore the problem of delivery and make that someone else's problem. And then I would work on DNA programming and someone smarter than me would figure out the delivery aspect and then we could do all that cool stuff later. Luckily, actually, though, when I got to Ron Weiss's lab and started interacting with Ron at MIT, we started talking about this new idea that, there was this new company that had just come out of stealth called Moderna. There was a German company called BioNTech that they were developing these mRNA therapeutics. Now, this is like 2013. So these companies are young. A lot of people didn't believe in the technology, but to me, it made a lot of sense because mRNA doesn't need to be delivered to the nucleus. It's active in the cytoplasm. And so they were developing the tech for like all sorts of different things for cancer and this and that. But it became pretty clear to us when we looked at what modified RNAs could do, that the duration, the specificity, everything about a simple mRNA was really optimized for a vaccine and not very applicable outside of vaccines. So in order to develop mRNA beyond just a vaccine modality and into like a therapeutic, so it could actually express a therapeutic gene, you needed higher levels of expression, you needed higher levels of specificity, you needed higher levels of control in general, safety switches, all of these different tools. And we were a synthetic biology lab, so it was, gene circuits all the time. The problem then became that there was no gene circuit technology for mRNA. Basically, everything that had been made at the time was for DNA. There were all transcription factor and cofactor engineering, TET system, doxycycline, all this different stuff. None of it was really made to regulate messenger RNAs. And so that was a pretty cool PhD project to work on because it was very translatable. It was on a modality that I had a lot of confidence in, and it basically was like a bottoms-up engineering approach. And so we started working on that. That's when I met my co-founder, who was a postdoc in the lab. He convinced me about like all of these different things that we could do. And then we, started the work. We were pretty successful with it. We published some papers on it. And then, the company sort of came later.
Yeah, that's so awesome to hear. Did you know that you wanted to be an entrepreneur when you first started your PhD?
No, I'm like most people, I think I wanted to be a professor.
Mm-hmm.
I think, and I don't know what things are like now, obviously, with things like Nucleate and the MIT Biotech Group and other biotech clubs and founder-led biotech, and like things are rapidly changing in the culture of biotech graduate school, I think. But when I entered, I remember probably most of my class wanted to be professors. I think maybe one or two people out of the 23 are. And so, at the time, I wanted to be a professor, but I thought being a professor was like, I wanted to be a professor so I could like do research and spin it out into companies, which is kind of like the entrepreneurial base case of what was sold to us at the time, like, for scientists. The ecosystem changed while I was in grad school. And part of that was led by like what we were doing in grad school, right? We founded the MIT Biotech Group in like 2014, and then we started building other things and all this other stuff came forward, and we, venture creation got more popular, and we got to see more of that, and I got to get exposure to, what you could do with a company. And I think that the market and sort of the access to capital changed things, where we saw this transition from like companies being just strictly development corporations that do research just to develop assets, more to where companies were doing fundamental, interesting, applied research on the hope of building a number of different therapeutics or technologies. And that was much more interesting to me. And then, so I think that the leverage that you get as a company, where the capital raises are different, and sort of the milestones are different. You get like people together to do like individual to do one big mission, and you can build large organizations around singular missions, is really powerful.
Yeah, that's incredible. Yeah, I think we'll move into our slides to talk a bit more about the paper, and then we'll get back to talking more about Strand and kind of the formation and evolution of Strand over its lifetime. So let's see. All right. So digging into some pre-work slides prepared by Bowen, he brought up kind of in thinking about the background, Jake, what you mentioned with, there was this issue with delivery, and then you were motivated to think very in depth about RNA. Could you give us a little bit more about, what makes RNA so special, and what makes RNA such a unique modality over DNA?
So I think it comes down to two things. The biggest part of it to me is about delivery, right? The nuclear membrane is very strong. It's very hard to pass through, right? The cellular membrane is very permeable, right? You're endocytosing things all the time, and sort of to trick the cellular membrane into taking up a liposome and then escaping that liposome inside of the cell is a much easier challenge than I think tricking the nuclear membrane, which is mostly made up of like, in terms of proteins and nucleic acids, it's all active transport, right? And so tricking the nuclear membrane and the nuclear pore release structures into importing foreign nucleic acids is really hard. I actually don't even know where that science has evolved in the last number of years. I left that behind in 2013, but it's still a very hard problem. I think the second problem, or the second aspect of it, outside of that, is there is a safety element, right? Which is that RNA, despite what a number of people on Facebook might say, RNA is not like integrating into your chromosome, and actually it's pretty much impossible, right? I mean, there's not really endogenous retrotranscription that's going to take an RNA transcript that you've transfected and, you know, retrotranscribe that into DNA and get it into the nucleus, at least not in anything that we know happens in any sort of frequency. And synthetic DNA is probably a low risk for that, but it's still like a risk. And so I think, and then if you compare it against like viral-based gene delivery, right, you have things like lenti, which is actually incorporation into the chromosome, and adeno-associated virus, AAV technology, which is like very long-term expression. And, you know, to me, it's like RNA sort of has like that sweet spot, especially if you can, you know, fix like redosing and toxicity issues. You can continually redose as well to keep levels for whatever period of time.
Yeah, absolutely. So with this technology, where, you know, as you said, you know, a lot of the focus was on DNA as opposed to RNA. Were there any challenges, any foreseeable challenges at the beginning of this project that you were most worried about?
I mean, yeah, like first of all, it's a huge pain in the ass to do RNA research, especially when you're in a synthetic biology lab and like everyone else in the lab gets to just like do a mini prep and be done, and have their DNA circuits, and you have to like do a mini prep and then do a linearization of the template and then do an in vitro transcription reaction and then purify that and then do like encapsulation stuff. And, you know, it's, I mean, that's just like fundamentally annoying. I shouldn't say, I think my scientists are around here, they're going to hear me complaining because they have to still do that. But, you know, like there's a lot of limitations of RNA at the time, right? First of all, like my biggest concern was like, we wouldn't be able to build technologies that could actually control the mRNA, right? And so the issue there is like, no one really knew how to control messenger RNAs, right? Your body mostly just degrades messenger RNAs, you know, and a lot of times it does it in like a very non-specific process, right? There's very few like completely, the microRNAs are the closest thing to sequence specific, and then they're obviously very promiscuous. And so you have this broad scale silencing of messenger RNAs that happen intracellularly. And can we really make like specific, because if you're going to regulate an mRNA transcript, you need it to be very specific in that regulation. Because if you start introducing RNA regulation technology into a cell and it's promiscuous and it binds like, you know, an essential protein transcript, then you're going to kill that cell. And we saw that with some of the things that we tested throughout time. So that was definitely like, I mean, and the other thing was like, well, how are we going to even regulate transcription or translation, right? And we started with degradation as well, microRNAs and deadenylases fused to RNA binding proteins and things like that. But then we moved on to this idea of, can we bind the 5' UTR and sterically hinder translation? And can we bind the 5' UTR and you know, even make these proteins so that they could sequester the mRNAs to like translationally null regions of the cytoplasm. But I mean, you know, early on, the big concern was like, is this even possible? Did I just open up my PhD to like a fundamentally impossible problem?
Yeah, absolutely. That's always the fear in starting a PhD project for sure. I love that you brought up localization. That's something that maybe I missed it in the paper, but didn't come up, but something I think I was continuously, you know, kind of curious about. I love thinking about the cell as a huge place. Can you comment on that a little bit more?
Yeah, it's in this paper, though it's probably undersold by the time the editors and reviewers got their hands on this thing. I thought it was very interesting. I don't know if we were told a number of times by other journals that actually mRNA therapeutics are not high impact enough for publication. So maybe I should have held off on publishing this until 2021. One of the things that's in this paper, you might see there's a protein regulator called TET-R DDX6. So what we wanted to do was hijack the TET-R system that's used for doxycycline dependent binding to the TET operator. And you can actually use that to bind a aptamer that Jacqueline Niles' group actually had elucidated. So a grad student by the name of Stephen Niles used the SELECT system. He found an aptamer of mRNA that binds to TET-R in a doxycycline dependent manner. We actually used that in the paper for one of the small molecules that's responsive. However, what we found was like TET-R itself was not very good at blocking the ribosome. So different proteins binding the 5' UTR have different dynamics of ribosomal blockage. And so by not being able to block it, it became, it was great that it was small molecule dependent, and it had actually a different inverted logic from the destabilization domain logic, right? Which was small molecule on versus small molecule off. And so we really wanted to use it. And what we found was these dead box helicase 6 proteins, DDX6. What they do is they localize to P-bodies within the cells, which are translationally null region. They're in a region of the cell that block translation for whatever reason. And what we found is if we fuse TEDR DDX6 to or if we fuse TEDR to DDX6, then when the TEDR bound to the RNA transcript, it would sequester the entire transcript with itself into these P-bodies. And what's interesting, I don't think this is in the paper, but what we also found was that localization has different effects in different cell types. So for instance, it doesn't work very well in muscle cells. And that was really annoying when we were also looking at vaccines. But what we found was that muscle cells, since they're like protein factories, have no P-bodies because there's no reason to suppress translation. They're like these translation machines. So so that was actually kind of cool as a pseudo I guess like positive control where it didn't work as well in muscle cells. And then we found out they don't have P-bodies in that.
Yeah, that's fascinating. I'd love to also open it up to questions from our Nucleate Insights cohort. Or Bowen, if you have any questions that you thought, or things that you were particularly excited about with this concept.
Yeah, I don't have current question, but I will have question like later. I already read in Stack actually. Yeah.
Okay, no worries, no worries.
Sure.
Okay, yeah. Well, I'm going to move on to the next slide. Yeah, Jake, if you don't mind, could you walk us through the concept for the on, off, and 2 output switches?
Absolutely. Yeah. So, all right, let's start here. So the basic thought here is that, like we said, the RNA binding protein, and I don't have a cursor. So if we look to the left, you'll see L7AE is produced from this first transcript, right? Now L7AE binds to this K-turn motif in the 5' UTR. And so when it binds that motif, it essentially is going to block the ribosome from scanning from the way that mRNAs get translated, right? Is that eukaryotic initiation factor complex basically takes the cap and the poly A tail and circularizes the mRNA, and then a ribosome binds on and it scans until it finds a start codon. And so what we can do is by binding L7AE, which is an RNA binding protein that's derived from bacteriophages, and we can bake its binding motif in upstream of our gene of interest, which here is M. venus as a reporter. And as L7AE binds there, it will block that ribosome from being able to access, from being able to get to the start codon. Basically, as it gets to L7AE, it just can't continue scanning and it falls off the transcript. But so the way this works with small molecules is that a destabilization domain is fused to that to the L7AE, and so TMP, which is an FDA-approved small molecule, so when TMP is added, it stabilizes the destabilization domain, which then allows L7AE to bind the K-turn motif. But without the small molecule, the destabilization domain actually makes the entire protein very unstable, and it gets degraded by ubiquitination and just general protein degradation machinery. And so the reason that this is important, right, is that in general, the way we wanted to build these switches was, we knew destabilization domain and these small molecule technologies worked for regulating protein stability and regulating protein availability. But the problem is that they require a covalent modification of the protein, right? So if you put a destabilization domain on M. venus itself, or, say you're down the line and you're like at our company now, where you have an active protein that you want to do something biologically, if you're trying to regulate that, but you're covalently modifying it, there's been no shortage of clinical trial data that has shown that by covalently modifying the protein itself, you totally screw up the biology of those proteins. And so we want to have a way to actually inadvertently control the translation of the protein rather than control the protein levels directly through that binding. So essentially, when TMP is added, that's how that will turn off the M venus expression. And so that's what you want to be able to see in that first switch. In the on switch, it's a different sort of small molecule system. So TET-R is being utilized here. So TET-R is going to bind to an RNA aptamer in the 5' UTR. And the way it's going to bind is, and the way that the protein works is that in the presence of doxycycline, TET-R actually binds doxycycline and changes its conformation so that it cannot bind nucleic acid. And so when the doxycycline is present, the TET-R is released. And so now you have the opposite logic where the small molecule presence is inhibiting the repression and inhibiting the repression of the downstream transcript itself. And now finally, as you get to the two output switch, essentially, this is just much more complicated where TET-R is regulating L7AE, and then L7AE is regulating the M venus downstream. And so in order to actually get the M venus to be expressed, you need TMP to not be present because that will cause L7AE to stabilize and repress it. But you need doxycycline to be present because you need to relieve the repression on L7AE downstream. And so this is really just like an exercise in getting a paper into a Nature journal. Just kind of pushing the technology to the next step, just to showcase fanciness.
Yeah, absolutely. Were there particular applications of the two output switch that you were excited about or saw potential for?
Yeah, and actually, when we were trying to get this into like nature, we actually were trying and just eventually we're running out of time with wanting to actually adapt the technology into a real company. But we were looking at some muscle differentiation work actually. So for instance, with like undifferentiated muscle cells, and if you might want to push them, along some like myofiber differentiation, for instance, in alleviating some of the symptoms of DMD, Duchenne's muscular dystrophy. There's some differentiation that you can do with where you want to have the expression of one gene, and then you want to switch and express a different gene. So what we wanted to do is express AKT, which is this master regulator protein, but only for a small amount of time. And then we wanted to switch it over into follistatin, which just increases the AKT. If it's on for too long, it's a master regulator and it actually gets very toxic. So you only want a short burst of expression, whereas then you want to switch on the follistatin, which kind of really beefs up the muscle cells. And so we were working on that. It was starting to look really good, but it was incredibly tedious to assay that and just very annoying. And we just couldn't get it done.
Yeah, no, that's so fascinating. I think I'm coming from a circadian rhythm background, and I think there are so many instances where you have this burst, this translational burst that's really important that you see all the time in normal physiology that could certainly have potential in therapeutics. I think moving forward, then we go on to thinking about the Replicon. What motivated thinking about both the mRNA as well as the Replicon? And how was this idea kind of formed?
So, the technology that we wanted to build, and this continues to Strand today, we want to build technology that's broadly applicable to mRNA molecules, right? The mRNA molecule itself, whether that's modified, replicon, circular, anything else, non-coding even, right? You want something that at the end of the day is able to control and modulate the mRNA molecule. So we had these two different pieces of technology, and the Replicon was honestly what we were more excited about, to be honest, right? It was what we're developing here at Strand in a more diligent manner as well, because Replicons are capable of, expressing more protein. They're capable of, expressing for longer durations. And also what was interesting about the replicon is that so the replicon here, you can see the non-structural proteins on the left. That's actually that's actually the majority of the sequence. It's just abbreviated here. But then there's the SGPs, which are these subgenomic promoter regions. And so in the normal replicon, there's a singular SGP in it. And in the alpha virus, which the sequences are derived from, the SGP actually drives the expression of the envelope proteins. But we wanted to, obviously, in a replicon, you knock out the envelope proteins, you replace them with your cargo. And what we also found was that by recapitulating certain other elements of the replicon into, so replicating the subgenomic promoter and replicating the 3' UTRs all into these parent constructs, we could actually make independent expressing units of the same transcript of mRNA, because when this is replicated, the replicase is binding the subgenomic promoter, and it's using that to actually copy the RNA. And so you're getting actually all these different mRNAs, you're getting, all different aspects created inside the cell. And it's a very creative way to get these complex circuits into cells, because they're all on a single transcript, versus modified RNA circuits, which are multiple different modified RNAs that you have to then encapsulate into the same particles. And there's all sorts of issues around that. So so that was really sort of the two things that were driving it. And then the ability to, recapitulate these circuits is what we wanted to be able to build.
Mm-hmm. Yeah, that's incredible. Were there aspects of this technology that you felt were particularly novel to your project that were exciting to you?
Yeah, so I mean, the replication of or the ability to duplicate the subgenomic promoters, and this discovery about being able to move the 3' UTRs upstream of all these subgenomic promoters, and how that actually increases the expression from those regions. So, like L7A, you would see that like the closest to the 5' UTR gene has the lowest expression, but you can rescue a lot of that expression by including the 3' UTR up there. And so, just the general architecture and I mean, in any paper, there's about 1 billion experiments that go into that final piece. But I mean, there's how do you incorporate like the how do you incorporate these aptamers into the subgenomic promoter? How do you build these different subgenomic promoters? We also found that like we could vary the size of the subgenomic promoter and truncate it, which actually changed the expression dynamics, and the expression dynamics changed the entire performance of the circuit. And so, yeah, there was a lot of that.
Yeah, that's awesome. Are there any questions from the group? And also, Bowen, if you want to jump in with any questions.
Yeah, I just when I look into the system, actually I have two questions. First is actually because the mRNA currently inside this replicon or the mod RNA also all contain a lot of loops, and usually people consider a loop of RNA would induce the innate inflammatory behavior of the cell and lead to toxicity. Yeah. So what's your vision on this part? Because your current, the design in current paper always use loops as a control element. Is there other possible way to also use a similar control element without a loop, for example?
Yeah, we're actually looking into that now at the company. I can't get too much into the details there because of things, but so we never really found that the loops were inducing immunogenicity as much as the replicon itself. So especially in these earlier primitive versions of the replicon that we were using when we were in academia before we built this design engine here at the company, replicons themselves are immunogenic. So a lot of this work was actually done in BHK cells, baby hamster kidney cells, because they're actually deficient in interferon. So they're easier to sort of really explore without solving the immunogenicity issues. The immunogenicity issues are what we spent like the first year as a company actually really working on in terms of translation. But the replicating RNAs themselves, loops aside, and aptamers aside, you actually create a lot of immunogenicity because it's a viral sequence, right? It's a viral sequence and it's replicating. So as it replicates, it forms double-stranded RNA, which is much worse than loops in terms of pattern recognition receptor excitation, right? And so we never really parsed out like if the loops were also causing issues. Our modified RNAs are super well tolerated, even with these loops. And we, have seen like in vivo regulation as well. So that was, maybe not an issue that we ran into at that point.
Yeah, thank you. Yeah, this answer very good. Actually, also my second question is about the replication. The replicons toxicity. You already answered it somehow.
Yeah.
Thank you very much.
Yeah, no, that's a great question though. I mean, and there's a lot to think about there because, replicating RNAs, like we've done a lot of work now as a company, actually building up a database of different replicating RNA sequences, different types of replicating RNAs, as well as different variants of the ones we have internally, because we can actually find that by making different mutations in the replicase actually allows us to alter the immunogenicity. And we believe that's mostly from altering both the immunogenic motifs, like of the replicase itself, but also of changing the replication frequency, right? So if you reduce the replication rate, you can actually reduce the immunogenicity. And there's sort of a near-term maximum where you reduce the immunogenicity so you get more expression before you reduce it too far where you get less expression, right? So there's sort of like a maximum in there that we found for a number of these species as well.
Thank you very much. Yeah.
Yeah, that's fascinating. I think, Stefan, you had a question?
Yeah, thanks very much, Rebecca and Jacob. So do you consider any other small molecule controllers besides DOCs? Like, for example, could you eventually make a circuit where it has something like a trace vitamin or mineral to control something like this? Thanks.
Yeah, so we actually used a couple other small molecules that didn't make it into this paper. I don't think. The two other ones that we looked at were 4' OHT, which actually they may be in the SI. I don't really remember when we were fully writing this, but 4' OHT and also Shield 1, which had destabilization domains that were primed to them. However, one of the things that was kind of exciting that emerged during the time that we were designing these circuits was someone in George Church's lab produced a paper that claimed that, within like 8 weeks or so, they could design a destabilization domain to, any small molecule metabolite or something. I think they did it for like progesterone and maybe one other, like some hormones or something. So, theoretically, you could do that for the destabilization domain circuits, right? TED-R is like, sort of like, it's specific.
Yeah, that's awesome. One question that kind of came up in the group beforehand was also about photoregulation. Can you kind of give a little insight into small molecule versus something like photoregulation?
Yeah, photoregulation was something I was actually really interested in, and there was a postdoc in Ron's lab at the time who actually now works at Strand, who was working on some cool like blue light receptor protein circuits that I thought were really cool, and I wanted to incorporate them. But as you guys probably know, there's always like 30 different projects that you can like think about and ways to take your PhD that you just like never get around to. I see one of the cooler things about running a company that's based off your PhD research is like, some of those projects now we have entire teams devoted to solving them, which is really cool. But photoregulation is not something we've looped back around on just yet. But there's no reason that like, if you could build a system, with these regulators, you should be able to recapitulate it. I haven't seen any of that work. I don't know if anyone in Ron's lab is really pushing this technology forward in this sort of way anymore, since we launched the company and took most of those people.
Yeah, it's awesome. All right, I think we'll move forward into validation.
Question.
Oh, sorry.
Sam,
Sorry, go ahead.
Sam Kessel.
Oh, Sam, go ahead.
Oh, thanks. I was just saying thank you, Jacob, for taking the time today, and appreciate for including me in this. So I'm a physician by training, so I'm very interested in how we're thinking about this in terms of going into patients, and specifically as it relates to like this specific slide. And one thing you mentioned kind of in passing is about duration. So how long this lasts for, I think is a really important point, because like when we're thinking about current gene therapies, they're a one-time dose, you're done. For this, one of the things I was trying to understand looking at this paper and then looking at what Strand's website is the duration piece. And it wasn't really, maybe I missed it, but in terms of how long can this realistically last for in a patient, I don't think you could answer this in this paper, right? Because it wasn't in humans, but when we're thinking about duration, would this, and then we're thinking about translating this into a therapy, would this be something like an injection that someone gets like once a month? Or is this something that, you know, you have one injection and you're done? In terms of small molecules, there are like multiple ways that small molecules can be used, whether it was to turn on the circuit or turn off the circuit. So this is something that you're imagining in your head is like, oh, you get a one-time, you know, injection, and then you take a small molecule maybe once in a while when you need to stop a dose, or like you need to get lab testing. Like there's a lot of complications here, and I'm very curious in how you're thinking about envisioning this into a therapy.
Yeah, that's a great question. So, I mean, I think when you translate this technology into actual therapeutics, you need to like kind of find like product market fit basically, or product disease, or, you know, technology disease fit. So where we're starting right now is on, you know, I use the term, this is a little controversial according to Twitter, but I use the term gene therapy to mean anytime you deliver a gene therapeutically into a cell, and genes can be carried on DNA or mRNA. So technically, like, yeah, the COVID vaccines, I think, fall in this like weird gray zone where I do think they're at least a genetic based therapy, where you're delivering a gene into a cell and it's causing a therapeutic response. Now, where Strand is really focused on developing a lot of this technology is in diseases where, you know, where 1 to 2 bursts of expression, right, from these vectors can actually have a therapeutic input. So our first drug that's going into phase 1 clinical trials next year is an IO drug for solid tumor immunotherapy. So there's a follow-on paper that Daryl Irvine's lab, along with Ron, put out a little after we finished up this work, which we have now commercialized into our first drug. And essentially the thing there is like, you know, can you use this technology to get into a tumor and then cause the tumor to express whatever you have, whatever gene that you have encoded on it? And the advantage here is like, you know, all sorts of protein therapeutics have been administered into patients, into the, you know, into the systemic, into the bloodstream, right? IV dosing. And you have this issue where like, you know, you either have bad localization to the tumor, or you inject the protein into the tumor and it diffuses or it degrades very fast. And so the value here is that if you can turn the tumor into a little protein factory that's constantly producing like an IL-12, for instance, which is what our first drug expresses, then you have like this ultra high local concentration in the tumor, which gives you a good immune response, but very low systemic toxicity because the, you know, whatever is diffusing out, you can sort of like modify that. And so that's really how this mRNA technology gets translated into a therapeutic. Of course, like now small molecules, and also if you read the previous paper to this, which is the Nature Biotech paper on the first generation of the circuits, what you'll see with that is like, you know, it's all about being able to control messenger RNAs for specificity, right? Small molecules give you a potential to design things like kill switches if you're going to express something that's immunoinflammatory. And then the microRNA-based expressions circuit cell type specific expression system that we built in that first paper really allows you to like, you know, control the cell type that the mRNA is going to express in. So like if it goes into the liver, it won't express there, for instance. Does that make sense?
Definitely. I'll say, and I know we're short on time, but I was curious, like, is it hypoxia or what is it in the tumor that is specific that's driving the selection?
So for tumors, for our first drugs, for tumor specific expression, it's more focused on microRNA based switching, right? So microRNAs are, you know, differentially expressed across all sorts of tissues, both can't like, you know, basically whenever your cells differentiate, they change their gene expression. And part of that is changing microRNAs, which regulate endogenous genes. And so you can actually take the data and the differential microRNA expression and use it to actuate your mRNA. So if you go back, it's not in this paper, of course, but like in the 2015 Nature Biotech paper, it shows sort of the ability to use those microRNAs to control the cell type specific expression of messenger RNAs. And so that's really what's driving that. The small molecule technology has actually not been like intensely incorporated into our current pipeline, though it's sort of under development for things like safety switches.
Yeah, that's great. I think moving on to the validation of the system, were there things that were unexpected that you came across in validating your on-off and to output switches?
Yeah, I mean, some of it didn't make the paper. For instance, we found that like, some of our other regulators, which were actually degrading the messenger RNA or like, using things that were like endonucleases, we found that they were very bad at cutting modified mRNAs. The modified nucleotides actually inhibit the ability of like Cy4 to cut the messenger RNA itself. And so, that sort of limits like what you can do for modified RNA circuits in particular. I'd say, for this particular data, there wasn't like anything super, by the time we got here, like there wasn't anything like super, unknown. I think that the TEDR DDX6 system was pretty cool because of how sort of like uniform the response is, very, very binary. And I think that's because, the steric hindrance is sort of a different, it's not nearly as tight. There's more leakiness of the ribosome blasting through the protein. Whereas like if you sequester the mRNAs into P bodies, then they sort of just shut off.
Yeah, wow, that's cool. I think our conversation has already started to move toward thinking about application of this technology. So we'll go ahead and move on. So I think, we've already brought up applications like cancer therapeutics, which is awesome. How did you decide on that particular application?
So at Strand, we have this philosophy that we want to make things as big of engineering problems as they can be versus biological problems. And so for cancer, for immuno-oncology, actually, in particular, you have a lot of like known mechanisms, right? IL-12, they've known from the 90s, right? That this is a good stimulator of the immune system, even before they had checkpoint blockade. And so the issue with most IL-12s is like, how do you increase the circulation? How do you increase the targeting? How do you mask it so you don't have off-target, systemic toxicities? All of those things that people have tried to solve mostly by protein engineering, right? And so, it opened up a thing where it's like, well, we know that this protein has a therapeutic effect. Now we just have to like basically turn it into an engineering problem where it's, well, how do we get a tight, localized, long-term expression from a tumor? And have some localization of the protein, and there's some other engineering things, but it's not taking nearly as much risk on like developing of a new, therapeutic modality itself, right? Or even some rare diseases, there's some hypotheses of like, well, for this disease, if you delivered this protein, then you would fix the disease or sort of help. But of course, like that also opens up the door of like, well, where's, how harsh is the regulatory environment going to be to get a new platform in? Whereas with cancer, we can both, have what we think is a very promising drug while also having like a regulatory burden that's just a little bit lower. So it allows us to move a little more nimbly.
Yeah, that's great. I think Stefan has a question.
Yeah, thanks. So speaking of cancer, it seems like this modality is really geared towards sort of a non-inherited condition such as that. So for something like cystic fibrosis or Huntington's, where you have a genetic component that's in the code itself, could you see a system like this eventually becoming integrated into the host genome to sort of fix stuff like that, maybe even in a controlled fashion?
So I think with that, it opens up sort of a different sort of modality, right? So like cystic fibrosis, if you have like single mutation, right, in the gene, what you can do is you can use a system like this to actually do like targeted cell type editing. If you want to just precisely edit like individual cells, because if you use some of the cell type specific technology that we have as well, you can actually build mRNAs that are going to deliver like a genome editing, like a Cas9 or something that's going to then be cell type specific. And so that sort of opens that up. I think that mRNA itself is not going to be a vector that gets integrated into the genome because again, like the mRNA construct would have to be retrotranscribed. You'd have to also deliver DNA, right? And then you have like these issues of like, can you actually get this into the nucleus? With Cas9, you have the advantage, right, of delivering it in messenger RNA, translating the Cas9 with a nuclear localization signal in the cytoplasm, which then binds to a guide RNA, which is also in the cytoplasm. And then the NLS will actually active transport the Cas9 into the nucleus itself. And so that sort of gives you a better advantage to actually do any sorts of those things. But, if you want to be making some of these, there's some gene editing, I think, approaches where you want to control like the level of Cas9, and you want to control the cell type specificity of Cas9, and you can run that via, this sort of technology.
Thanks.
That's great. I think Will also has a question.
Sure, it actually dovetails with what you were just talking about. First of all, thanks. I'm a first-year biology PhD student, so it was really nice to hear about your evolution of your PhD work. Yeah, so I guess some concern with sort of Cas9 gene therapies is with AAVs and things like that. With genetic disease, you want like more of a constitutive dosing throughout the lifetime of a patient potentially. But with AAVs, you have like a cargo limit. Have you looked into, or has anybody really like done a quality systematic analysis of like how big of a cargo can you deliver with modified RNAs?
Yeah, I mean, with modified RNAs or replicating RNAs, like you have your limitation ends up being, especially for replicating RNAs, more so on your manufacturing than it does on your on the cargo, right? So if you look at like this paper, for instance, like the Replicon circuitry that we put on, that's a single cargo of like 4 different proteins with regulatory machinery that's all encoded. It's like, I think like 6,000 or 7,000 base pairs worth of protein that's being produced from these single molecules. Modified RNAs have, other sorts of issues, but for the most part, like you can deliver most proteins, but your expression just ends up being low always, right? The expression for modified RNAs in general is always low. That's why it's limited mostly to like antigens for vaccines. But with any of them, it's going to be that as you increase the cargo, especially replicating RNAs, you increase the length of the mRNA transcript, and you have to manufacture that. And you have to manufacture it through an in vitro transcription reaction. And the thing about in vitro transcription reactions, and actually is what makes replicating RNAs themselves, like even with a basic cargo, much harder to manufacture than a modified RNA, because they're about 5 to 10 times as long. T7 polymerase, which is what's used to basically copy a DNA transcript or a DNA template into the mRNA transcript, T7 is going to fall off of the DNA at a given rate. And as you increase the length of the transcript, that likelihood that it's going to fall off given a given fidelity rate is going to increase. And so you can actually see this with like fragment analyzer data. You can look and see that like as the length increases, the early termination products increase over time, and then you get some like full length material. And I think like even in most CDMOs that are in the replicating RNA space, they're able to manufacture like 35 to 40% full length material, right? So your final RNA product is like 35 to 40% full length. Well, we've done a lot of work here to actually get that up much higher, now breaking 90% full length. But it's that's probably the bigger problem than like the RNA molecule itself.
I see. Thanks a lot.
Yeah. That's great. I think we'll start. Oh, sorry, Will, did you have another question? No, okay. I think we'll go ahead and start moving the conversation to thinking more about Strand. Jake, what was the point kind of throughout this project when you first recognized that you had a product or you had an idea that may have been, quote unquote, worthy of becoming a company?
So it's my co-founder who actually, I think, saw it more than me. I was looking at venture and some other things to do and thinking about what it would be like to start a company in the future. And the thing was really that we were looking actually at immuno-oncology as an area, and then thinking about like the growing opportunities of what messenger RNA could do, looking at what like Moderna was trying to do, for instance, and seeing that like at the end of the day, the fundamental shortcomings of messenger RNA, which were, duration and specificity mostly, could be solved by like a lot of the technology that we built. And it's not necessarily just in this paper. This paper had a lot of the Replicon work that we did and the different engineering that we could do to that. And the previous paper had a lot of the mod RNA circuits and some of the cell type specificity that we're able to achieve. And so what we wanted to be able to do was, we basically saw, okay, there's a lot of people trying to solve IO by cytokines, right? Can we get cytokines in these in these cells? Can we pegylate them to increase their circulation? Can we mask them to make them less immunogenic as they circulate through the bloodstream? And any other thing. And to us, it was just like, well, if we can express at high levels with the replicon and we can express at specific cell types with this programming technology, then essentially we can deliver these messenger RNAs into the tumors, either intratumorally or systemically, and then create those proteins directly inside of the tumors. And it sort of like opened up a world of possibilities that seemed like, well, this is kind of what everyone's trying to do. We can actually do it with this technology. Mm-hmm. That's what led us to jump off into that area.
Yeah, absolutely. What was the first thing that you did towards starting Strand?
Oh, early on, I think you got to kind of start to develop a roadmap of like, what the technology can do today, what you want it to do in the near future, and sort of the steps to get there. And as a grad student, then you need to like talk to as many people as possible, both about that plan and also like trying to meet people that could potentially serve as advisors. I think Nucleate's obviously got a wonderful program to help with that. That's why I've been an advisor of Nucleate for a number of years. I think it's really powerful and wasn't a tool that I had back then. But having, really recruiting people who can sort of help you develop the idea, you need to take from like, your scientific perspective, which is like, how do we build a paper, to like, how do we build a strategy towards a commercial product or towards a clinical product? And that's like really the first thing. And then you just have to like start meeting, you need to incorporate, you need to do all like the dumb, boring things. But then as you start to build that, as you start to get a strategy around like getting your own IP, then you have to like build a strategy around like, okay, well, how do we now pressure test this idea of here's our plan, here's our pitch, let's get this in front of some investors, and the timing of that and when you're ready to actually present to investors is there's an art form to that too. I think I think most of the teams I've worked with in Nucleate, I spend most of the time trying to get them to make like an actual pitch deck that's not a science deck. That's also what I did when I was starting. It was mostly a science deck, and we didn't get funded at that point. So, wrong move, big fuck up. But, we were definitely able to like pivot and adapt and build better decks and find investors that saw the potential in what we were building and sort of backed us. I think that the world has also changed a little bit now in terms of like the capital that is available for founders in biotech versus where it was maybe like 4 years ago.
Mm-hmm. Yeah, it's cool to hear. I think those early hurdles lead to resilience in the long run. I think that that'll be awesome to see. Were there any particular technological or proof point milestones that you had to pass on the science side before moving towards securing funding?
Yeah, actually, we got kind of lucky because we were able to convince. I was able to convince Daryl Irvine is one of our co-founders, to like start working. So we knew our technology could do this, and we wanted it to work in tumors, and we didn't have like that data. We knew that it was the right fit, but we didn't have like specific data on it. And so, we were able to convince Daryl and one of his postdocs to start kind of working on that. And they were able to generate some data around like some early response rates to, this technology and how it works and yada, yada, yada. And that sort of like strengthened our pitch overall. It's definitely, so I would not have done it if I had to go back and do it or advise myself, 4 or 5 years ago, I definitely would have not done it the way that I did. It's probably more effective to like go in with like a very tight pitch story, and think more, think less like a scientist and where you're presenting like a whole bunch of data and sort of talking about like, you really need to lay out like what you're doing and a mission and make it very clear. I think one thing that I tell a lot of would-be founders is you have to make someone give a shit about your science before you tell them about your science, right? Like even if they took the meeting with you, even if someone got them really jazzed, like the beginning of the meeting has to be all about like, why should you care about this? Because otherwise, like, people see like a million things a day. They just won't pay attention. They'll start answering their emails, especially in virtual world.
Yeah, totally. I think that that's a great transition to the next kind of part of our conversation, talking more about Strand as a company. I think so many, all of us really are motivated by clinical unmet needs and things like that. So, what were the initial clinical motivation that you saw with Strand, and how did you initially kind of create that catch or that early pitch?
Yeah, so in order to capture, I mean, the early pitch was shit. Like, I'll be very honest. It's very, very bad pitch. I've seen the deck. It's awful. It's embarrassing. I think the problem is that, like, especially when you work on this technology for so long, you get very excited about it. And it's all very obvious to you when you're the fact, like, you have the vision and you're like, you sort of like, to me, it was like, well, look at all these things we can do. Isn't that amazing? Think of what we could do. And I think that the trick is, like, to first get someone to understand what your vision is in the first place, get them excited about that vision, and then explain why you can execute on that given your technology. And that's really the art of, like, changing the thought process, because I see this in science presentations, even from great academics. Sometimes they get so caught up in talking only to people who are working on the same problems, where they lose, like, motivating it, because they know that, like, the people that they talk to, they get in front of them and they're like, oh man, what if a switch was more like digital than analog? Like, look at this synthetic biology switch. And then, like, boom, boom, think about that. And everyone else has already bought into, like, the thesis that that would be a good thing. And so you don't think about, like, motivating why that matters. And one of the hard things to do for, like, scientists coming out of academia, myself included, is, like, taking things that you spent, like, years on and putting them in an appendix slide, because no one really gives a shit about, like, that particular piece of data. And understanding that, like, there are things that are more important to you as the scientist, and will actually be differentiation points to your success in the future, that just don't belong in a pitch at all. They just don't belong there. Right? No one cares. No one sees it. Some things you will see, and you're right. But, like, the other people don't see it, and you need to show them what's going to, like, get them excited about it. And, if they dig in, and they see the cool shit later.
Yeah, I think you're getting at, of all the things that you can do, what should you do question. And has that changed for you over time with Strand?
I have not become good, but I've become a lot better at telling this story. I think it really helps that I don't do any science anymore. It's much easier to be at 10,000 feet when you're really at 10,000 feet of, like, what all the actual talented people here are doing. I think that as that happens, sort of allows me to have a broader view and get very excited about the science as, like, as a CEO. What you really are is a liaison between capital markets and capital expenditure, which is, like, your research team, basically. And so the reason that I think, how you're supposed to be straddling that is, taking the ideas from the research, projecting that to capital markets, and then getting capital market feedback and using that to inform some of what your research team is going to do, right? And really, like, sometimes the goal, the art there is, like, okay, like, yes, in a perfect world, we would de-risk X at, 6 months from now, but right now the investors need to see that so that they can believe that we can do that. We believe it, and we have things but we have to make it very obvious. So let's move that timeline up. Kind of straddling those worlds is something that you get better at with time. And I think it's really how, like, I do a lot of angel investing now, especially in biotech companies and in tech-enabled biotech companies. And I make most of those bets on if I think that, like, one of the founders or the founders themselves can grow into that role. Not that they will have it when they start, but there's, I have a thesis that is like, okay, I see, I work with someone over a few months and I can sort of see them growing in this direction, and the way they pick things up and the way that they think about communication, is something that I think is really promising.
Yeah, it's interesting. Are there any questions from the group on kind of the clinical aspects or the clinical challenges for Strand?
I actually have a
Yeah.
question from the slide. Oh, sorry, I didn't
Go
raise
for
my
it.
hand.
Yeah. No, that's fine.
Yeah, so I still didn't actually very get why Strand actually directly picked the solid tumor as a target. So this strategy seems very narrow and picked very specifically. Is there a special reason for that, or is because of the passion of the team? Yeah.
So I think that, again, solid tumors is a huge area, right? It's maybe one of the biggest areas in expenditures for medicine. And it also is an area that allows us to attack issues that we believe we can create engineering solutions for now when we have limited bandwidth and want to take, sort of tiered risk, right? Where we know if we can deliver in a certain way, then we can create therapeutics that have an actual biological response, clinical response in these patients. So solid tumors is really just like where we're starting. And it gives us like a very constrained environment to build solutions for in the near term, which are, multi-billion-dollar opportunities to put price tags on them, right? I mean, triple negative breast cancer in itself, just as triple negative breast cancer as a solid tumor is like a $3 billion a year run rate in capital expenditures, right? In terms of like total market size. So can we build solutions that have value, that entice investors in the near term, but also allow us to de-risk pieces of the platform that we can then turn on in the long term, right? So if you can develop manufacturing and formulation and delivery technologies for solid tumors, and then programming that allows you to switch off in non-target organs and on in tumors. Now, if you can then take all of that and pivot it towards T cells, for instance, instead of turning on in tumors, you now want to turn on in T cells, and all of the basic pieces still apply. Now you can maybe deliver a chimeric antigen receptor to a T cell and create like a in vivo CAR T approach. Maybe you can pivot this towards the kidneys and deliver something for like polycystic kidney disease or something like that. So all of that is sort of building up the platform, but doing it in like a very constrained fashion. It's very hard to take new technology, new engineering, new platform, and like obscure disease state and actually roll out a therapeutic there because there's like so many variables of risk.
Mm-hmm. Yeah, absolutely. I think Jess had a question.
Hey, thanks. Yeah. Hey, I'm Jess. I'm at UCSD right now in my PhD. Actually, Jacob, I had a question. I wondered if you could kind of go back to the discussion about pitching on a vision statement versus pitching on these kind of like details of the science. And I wondered if you could give us a taste or kind of a little example of, what are those statements that you would use and how would you kind of rope in investors within the first few minutes to say, hey, this is our vision and this is like a taste of what the technology is, but here are the metaphors that you can use to really understand what it's all about.
Yeah. So, I start my pitch deck with the vision of the company, which is, and the mission really of the company, which is to bring mRNA therapeutics beyond vaccines, right? So how are mRNA therapeutics going to actually deliver on all of their promise? And you sort of say like, we have this programming technology that we think is very important in the actual development of mRNA therapeutics in non-vaccine settings. And then you go from there to sort of talk about like, so like the current state of the art in mRNA, what are the shortcomings of regular modified RNA platforms? And you talk about longevity of expression, you talk about like specificity of expression, you talk about some other things. And then you sort of talk about why our technology, like the different aspects of how we approach those solutions. And then you get into sort of explaining the technology. So now you've talked about like, okay, if we want to really explode messenger RNA technology and actually access all these new areas beyond vaccines that apparently have so much potential value, then we're going to have to address these limitations of the current technology. We address them like this. And then if we can do this, it's huge. And then you go like, and here's like some highlights of why we can do that. Now, of course, like after the point of a first pitch meeting anyway is just to get someone to care, right? It's not, no one's going to come out of like your first hour long or 45-minute long meeting with someone and be like, I'm in, it's $5 million. Like what they're going to come out of it with is, okay, basically you want them to say big if true, and then spend the next few weeks figuring out if they believe it's true and if they believe that you're the person to bring that truth forward. And most people will say no. That's just a fact. you have to be very resilient to people telling you to fuck off.
No, it's good to hear. I think everybody in STEM has experienced failure, whether it's, you know, a paper rejection or grant rejection. And I think that hopefully that resilience translates over as well. That's cool. I want to kind of inch forward, just for the sake of time, to thinking a little bit more about the business model of Strand and how you came to kind of decide or make decisions when it comes to that. How did, throughout this process, how did you successfully manage creating and forming your team, and what are the things that you thought about early on about team formation when it comes to both the scientific side of things as well as the business side of things?
Yeah, I mean, we like to fit the team to milestones and then to find people that are smarter and better than us at, you know, the given areas to take on these problems, right? So, you know, early on, we knew that we needed to solve some manufacturing challenges. We needed to understand that. We needed to be able to do a lot of studies in animal systems, and people that understood immuno-oncology and could actually, like, you know, do more in-depth analysis of these different, you know, animal systems and these different, like, models of cancer, to really prove to ourselves that, like, the advances we were making with bringing, like, the platform forward in the first indication was actually working. And so, you know, our first hire, you know, the first person that we ever hired was an immuno-oncology, you know, sort of expert scientist. That was, you know, the first hire that we made was like, we need someone who has a skill that we don't have. Me and my co-founder are synthetic biologists, right? We understand the circuits and we understand molecular biology. We don't understand, like, animal models nearly as much, and, like, how to build, like, an animal research team. So we hired that first. Then we hired someone who had, like, a similar skill set as ours, because then when you're running a company, you can't really be working in lab. So those were our first two hires, and then started hiring some other auxiliary team heads, people with more experience in the industry, like a manufacturing head who could work on process development, then a lipid nanoparticle encapsulation head, and then building teams below those people to support their work. And so I think that you need to, like, sort of start with, like, the big milestone, which is, okay, we want to get to, like, a development. Like, for instance, I'll just talk about what we did. Like, okay, the milestone is we want to get a development candidate into IND enabling studies in these indications. We want to build a drug that we're confident to bring forward in the sequence and the formulation and the manufacturing, blah, 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 blah. So how do we get there, right? Okay, so first we need in vivo, we need in vitro development of the platform. We need in vivo, you know, validation of the efficacy and some baseline toxicity tolerance of the drug. We need manufacturing we need process to manufacture the mRNA, and we need formulation to encapsulate the mRNA and deliver it. And those are the big buckets you need to hire for. You need to hire people that are, you know, better and more experienced than you, and then build teams below those people over time. And then you also, you know, as you start to grow that and develop a platform, in order to get to the next round of financing, you'll also have to showcase that, you know, this isn't just one drug. This is actually has the ability to do all these other things. So it really, like, building the team is more so, in my mind, fitting to the milestones that you lay out. You need to lay out the milestones and then say, you know, how many people at what levels of what experience level are going to get us there? You know, how does this milestone get done? Break that down into, like, okay, we need to accomplish, like, X, Y, Z, 1, 2, 3, and then hire people to run those individual pieces, and hire people that are excited and about it.
Yeah, totally. Did you find that throughout designing these milestones that your story, you know, developed over time, over the course of your fundraise?
I think that our story developed a little bit over the course of the first fundraise. And I think big development was, like, the switch between that and the Series A. And then the Series A had a lot of, like, vision without nearly as much data behind the vision as we now have, right? Because you know, the first stage was answering, can we put a development candidate forward, and then we'll begin developing the platform more in depth. You know, one of the things that we didn't do, which I think was the right strategic decision, but it definitely made the, you know, fundraising and everything harder, was we really developed a first drug in a very diligent manner versus showing a lot of fancy pieces about our platform. Because what we wanted to do was, once we could develop a drug, then we can develop the infrastructure. At the same time, we were developing the infrastructure of the platform, but there's not so much explicit fanciness data to show. You know, the way that pays dividends now is that, like, we generate a shitload of very impressive and fancy data, because the infrastructure is so tight and so it's so productive, and you know, we just have like data like that just comes out of this like well-oiled machine. But those take investments. I don't think we necessarily did it 100% correct, but you know, it worked.
Yeah, I think sometimes with platform-based technologies, you see creation of the platform, then you know, really digging in on one indication, one product, and then kind of going back to think about the platform. Is that something that that you would change, you know, now that you are where you are?
Yeah, you know, I mean, the thing is, like, we weathered the storm and we got through, and we eventually were able to deliver on the promise that we thought. And like, the only thing that makes me hesitant on like the way that we developed the platform was the way that it was perceived by the investment community. And I think that, you know, if I was advising someone doing something similar, I might say, you know, in the seed stage, just make another milestone that is like some of this inflecting, right? It's not all about showing flashy technology so you can raise a shitload of money, but you do need to show that like part of this vision is de-risked. And I think that over time we've had the same vision for what's going to happen with the technology. We've just de-risked certain parts, and I think that we could have made like earlier rounds of financing easier on ourselves if we had done that earlier. However, that would have come at the cost of you know, the progress and the rate of development that we have for like our first drug.
Yeah, absolutely. Are there any questions from you all about the actual business model of Strand? If not, we'll push forward.
I had a question.
Yeah, totally.
You know, you mentioned the challenges associated with manufacturing, and I wonder how you all think about weighing the cost benefit of doing all that manufacturing in-house, or are there other companies that are working specifically on that manufacturing challenge that you would bring that, you know, obstacle out of house?
Yeah, so there are CDMOs, and in truth, we are leveraging CDMOs for outsourced manufacturing for GMP, because building out GMP is just so ridiculously expensive to do at this stage of a company. Long term, I believe that we need to own that. The difference is that, like, yeah, I mean, there are companies out there that have, you know, claimed to be doing these sorts of things for a long time. I've never really, you know, we've done side-by-sides of quality against like multi-billion-dollar valued CDMOs over what they can make of our product versus what we've been able to make with our custom process. And we just make better things. It's just a question in my mind of incentivization. There doesn't seem to be like a one-size-fits-all for any of this technology. And the market is too small for like replicating messenger RNAs, for instance, for a CDMO to really invest into the process development and on the timelines that we have invested, just because, like, you know, it's just not going to pay a dividend to them in the same way. Whereas like, you know, offering what they offer in terms of like effort to cash flow, they're just going to continue to do it the way they have.
Yeah, Sam has a question.
Yeah, so I spent a lot of time so I'm a consultant now. Spent a lot of time thinking about like different types of go-to-market strategy. So when you're thinking about going it alone, which seems to be the situation in the US anyway, you know, I know you have the partnership with Beijing, doing a co-development, co-promotion type of deal, or licensing, you know, especially at this early stage, are you, what I guess is your thoughts on, like, are you doing some combination of each? Are you thinking primarily going it alone? Like, because there's balance towards, you know, you get some initial, you know, capital for milestone payments, et cetera. If you do, you know, partnership type of agreement. So not sure where you're coming down on that. I'm assuming long-term you want to do a lot of go it alone, but interested to hear your thoughts on that.
Yeah, so, you know, you mentioned our partnership with Beijing, and, you know, they're a fantastic partner that does offer us a lot in terms of, like, you know, they they do a lot of oncology work. They've put tons of drugs in the clinic. And so they're a good partner because they're very knowledgeable. And also there is there's like some non-insignificant capital tied to that deal with them. So very excited about, working with them, though one of the things that gave us the benefit of is, we didn't want to take like too many chips off the table too early. We're very excited about what our first drug looks like in terms of like the deep, prolonged, great responses that we see from our drug. And of course, we're then building like next generation versions of that same drug to be, follow behind it in the clinic. And so we really want to own that as much as we can, especially given the fact that, like I said, that first drug was centered around like, we understand the biology and we're taking more of an engineering risk, and we are optimized to take engineering risk. And so we've built a lot of pieces around, being able to bring that drug forward. However, our long-term goal of bringing mRNA therapeutics into all sorts of tissues throughout the body and all sorts of diseases requires us to find partners that have more capital so that we can take, 20 shots on goal in the next 20 years, which we can't really finance. Like, it's very hard to finance that versus and also, not just capital, but someone who brings disease expertise, right? So if we want to go after like rare kidney disease, then we need a partner that ideally has capital to help us finance that effort, as well as has disease expertise so that we don't have to rebuild an entire, like all the different aspects of our team that we take through, right? Because the IND enabling studies for kidney disease are going to be, different. The clinical development of kidney disease is going to be different. And while we'll have people internally that'll be overseeing parts of that program, we definitely want to lean on the expertise of partners and also like use them as leverage points to take more shots on goal, knowing that, if we win any one of these, it's a great, piece of income for the company as well. But, in terms of like oncology, we want to take most of that alone for right now, just because like, I think we really know what we're doing there.
Yeah, that's great. I think some of this is related to a question that was submitted beforehand on, how much time or how do you manage exploration versus exploitation of a particular product? How much efforts are you putting into developing new products versus really focusing in on your main product?
Yeah, I mean, this is the problem that really hits seed companies hard, and we sort of definitely invested very heavily into our first product versus investing into our platform, which I think had pros, but definitely there's definitely cons, especially during the Series A fundraise and sort of investors that we probably missed because we hadn't de-risked part of our platform as much as we could have. I think that there's no set answer, and there's a number of different ways that you can do it and be successful, and then you could probably do it that same way and be unsuccessful. So it really has to be, and this is where someone who develops into, I think, a good CEO will sort of try to feel out that liaison that I was talking about, right? Because your job is to sort of understand capital markets and your company's position. And of course, you have a vision that you want the company to execute across. But in those early times when every single effort costs a non-zero part of your burn and of your capital expenditure, then you really have to sort of think about what is the opportunity cost that you are spending versus to get a certain piece of data, and how you're orienting your team to make sure that your next round data set is going to be palatable to the capital markets. And that can be very frustrating sometimes, because you don't want to spend all your time appeasing investors, because then you're not executing on your vision, you're executing on theirs. But you also can't just completely bury your head all the time and say, if we just do what I say, then that'll work. Because at the end of the day, what could very easily happen is you build something that's completely unpalatable to investors, even if it is great, and you end up in an area where the technology completely dies because there's just no support for it. That's just, I guess, the sticking point, and how you have to find your way around. If you can find deep pocketed investors that are willing to support you into the tens of millions of dollars to really execute your mission and then turn around and do something bigger at the end, you definitely do that too.
Yeah. Great. Jing has a question.
Yes, kind of similar like investor relationship. So I run a AI computation company for drug discovery, and it's the same thing. Similar things like when we develop like a technology that solve a certain protein design issue, and then the investor, because they talk to a lot of similar companies, they will come to us and say like, hey, I think another technology seems also popular, and we also should start working on, but we are seed stage. We are group of 5. So it's not really like practical for us to explore like a different products, even though like those things can be done like using similar computation technologies.
Yeah, I mean,
So I'm wondering whether
how do you deal with that, right? Like you're talking about your own investment. Yeah. So like, I mean, you should have like a good handle on why you are using your approach, right? And so the answer to any like, the answer to any approach like that from your investors is like, okay, well, like that technology does this and our technology does this. And we believe our technology in the, right now has the bigger potential, and we should be investing in it because we're going to be ahead at time X in the future. Now you can say that like, technology Y is still very interesting. And at the next stage, as we develop this infrastructure, we'll be able to also, start to explore this, and we'll actually be able to move more efficiently on this technology because we've already built the original infrastructure. However, we are placing our bet here where we are in a resource constrained environment. And also we're placing it here because we have a bigger belief in utilizing the technology this way. And sort of have those answers and be thinking about that other technology in the same way. You just have to be, and if the answer is like, if you don't have a good answer for that, then maybe it is something, there's no shame in pivoting, right? So having good answers on that front, I think is a good policy.
Yeah, great. I think we'll go ahead and move forward. We talked a little bit about competitive landscape. So I think pushing us to our final slide, are there any kind of, as you are looking ahead to starting clinical trials, are there any regulatory hurdles that you're anticipating and or that might be unique to your RNA product?
Yeah, I think that it's a new modality, so you need to prove out a number of things about it, right? Like, okay, replicating RNA in the body in general, what is that going to do? What are the concerns? What happens if this leaks out of the tumor and the RNA ends up in different areas of the body? How robust of expression? Like, will the IL-12 be overexpressed within the tumor and cause systemic immunogenicity? And all of that comes with, there are all sorts of IND enabling studies that are pretty much you have to do. Of course, we've hired a number of amazing people who are experts in sort of solving these issues. And so, you have to do these with like mouse and rat toxicity studies. And then non-human primate studies. We just finished our first non-human primate study last month, sort of showing that toxicity. And so I think continuing to show that data, that's where you sort of de-risk those questions. Yeah.
Absolutely. I think as we come up at the very end of our session, I want to be cognizant of time because I know you have a company happy hour, which just speaks to the probably good culture at Strand. Are there any, where do you see Strand in 20 years? And can you leave us with a final insight on kind of where you're at right now and then looking ahead?
Yeah, our goal is to be able to deliver mRNA into virtually any tissue within the body. Now, mRNA as a modality is not going to necessarily, at least in the near term, call that the next decade, be suitable for like, factor 8 for hemophilia or something where you're going to be like regularly dosing. But there are a number of diseases where you can reset homeostasis of a tissue by, duration expression, either, one time or every 6 months or every 12 months. And so if you can access these different tissues, perhaps you can actually reset homeostasis in a lot of diseases that are sort of have complex pathology. And so our goal is to enable that future by developing technology which allows us to get stable, low immunogenicity, messenger RNA for extended period durations with high specificity into the tissues throughout the body. And there's no reason that that's impossible, right? Because we know that every cell in the body has messenger RNA that it does use to express proteins. So from a first principles approach, there's only the very hard problem of why we can't do that synthetically right now to solve. So, in the next 20 years, in the next 10 years, I'd like to see us be able to express messenger RNA throughout the body in all sorts of different tissues in the, kidneys and spleen and immune cells and even neuronal cells in a long enough timeline and into the bone marrow and into, different skeletal muscular tissue to advocate or ameliorate like certain issues in the near term. And then 20 years into the future, can we actually master the fact of like dropping the immunogenicity of messenger RNA to a low enough level that even though we're having extended durations of expression, that we can actually do regular dosings that are tolerated in the same way a number of biologics are administered. And so I think having clinical programs and approved drugs in those areas and truly like mastering the future of messenger RNA is what we want to do and sort of the industry defining company that we're building.
Yeah, well, it'll be so exciting to watch. And thank you so much for taking the time to come to Nucleate Insights. And also thank you to all of you all who came out to join us and asked great questions. The people who came to our Nucleate Insight cohort, if they want, are they able to reach out to you via email if they have any further questions that didn't get answered?
Yeah, of course. I think you guys have my email and anyone can reach out to me. Sometimes I'm better at responding than others. Definitely reach out. And, of course, if people are interested in working on this sort of technology as well, we have a lot of open positions. So
Awesome. Well, thank you so much. I'll see you guys.
Thank you guys.
Have a great happy hour. Yeah.
Bye-bye.
Bye.